Okay, we'll be looking at HIV, right? The virus that causes AIDS. Okay, so AIDS is a disease with a progressive failure of the immune system because the HIV infects a type of immune cells known as the T helper cells. Okay, so T helper cells are lymphocytes or white blood cells. Okay, so what is the function of the T helper cells, right? So here I'm going to give you a very brief intro of the immune system, or next year you're going to learn it in much more details. Okay, so firstly, right, if we have the presence of an antigen in our body, antigens are substances that trigger the immune system, right? So they can be like structures found on, say, a virus or a bacterium, for example. Alright, so a type of cell known as the antigen presenting cells, or APC for short, would be able to detect the presence of the antigens, right, when we are infected. And it will be activated, which will then in turn activates the T helper cells here. Activated T helper cells would go on to activate the other immune cells for immune responses. Alright, so whether it is to kill a bacterium or to produce antibodies to bind to some viruses, all these will be done by the other cells, but they need to be activated by T helper cells. Okay, so T helper cells play a very important role in activating the other cells for immune responses when we have an infection. So when HIV infect the T helper cells and they die as a result, then that means that our immune system is not going to function, right? The other immune cells are not going to be able to be activated. All right. So, we have kind of talked about this already. Okay, then we move on to HIV, right? As a classification, HIV is classified as a retrovirus. Okay, so retroviruses, they are not just HIV, there are other viruses as well. All of them share similar characteristics. They all have a single-stranded RNA as their genome, and they have an enzyme called reverse transcriptase. Okay, so this enzyme, right, as the name suggests, it catalyzes reverse transcription. Okay, so transcription, as you have learned before, is when DNA is copied to RNA, right? So reverse transcription is the opposite. The RNA is used to be copied into DNA, right? So such viruses would copy their single-stranded RNA genome into DNA, and the DNA would then be integrated into the DNA of the host cell that these viruses infect. Okay, so later when we look at the reproductive cycle of HIV, we will see that this will happen. Okay, so let's take a look at the structure of HIV. Okay, as mentioned, it's a retrovirus, right? So it has single-stranded RNA as its genome, and there are two copies of it. Okay, so note the word I use, two copies. Right, so this is a bit different from influenza. Influenza, I use the word segments, right, whereby the 8 segments of RNA molecules are all different in influenza. But for HIV, it's two copies, meaning the two single-stranded RNA molecules, they are similar or even the same, right, if there's no mutations. Okay, so it's two copies, single-stranded linear RNA molecules. All right. And for the capsid code, similarly, right, it is formed by capsomeres, and the capsid code encloses the RNA genome. And within the capsid code, there are the presence of two other enzymes. One is reverse transcriptase, because it's a retrovirus. Another one is integrase. Okay, so reverse transcriptase is shown in this diagram you have in the notes as well. But integrase is not shown, right? So there should be another enzyme present within the capsid code. Okay, it is an enveloped virus, right? So there's the viral envelope. On the viral envelope, there are attachments of GP120 or inserted with GP120 as well as GP41. Okay, so GP120 is the one found here on top of GP41, right? So it looks like one single structure, but it is classified as two proteins, two glycoproteins, right? So GP120 and GP41. Okay, so next, the reproductive cycle. Okay, first step is attachment. GP120 is the glycoprotein that is responsible for binding to the specific receptor on the host cell. The specific receptor is known as a CD4 receptor. The host cell obviously is T helper cells, as we have mentioned before. There's also a secondary co-receptor called CCR5 co-receptor. Okay, so as you can see here, right, this is GP120, the one in blue and purple here. GP41 is the stalk, right, that is linked to the viral envelope. This is the CD4 receptor, and there's a CCR5 co-receptor here as well, right? So GP120 binds to both CD4 and CCR5, and that causes conformation change of the receptor, and that facilitates the entry. The entry of HIV is via a fusion between the viral envelope with the cell surface membrane of the host T helper cell. Okay, so because of the fusion of these two membranes, the nucleocapsid will be released into the cytoplasm. Okay, so this part again, I'm going to make a comparison or a contrast with influenza. Remember, influenza, the entry involves endocytosis. Okay, so the entire virus is placed into an endosome. Then the viral envelope fuses with the endosomal membrane. In this case, there's no endocytosis and there's no endosome either, right? The viral envelope immediately fuses with the cell surface membrane. Okay, so there's a difference there, yeah? So this diagram shows you what's happening, right? So when the GP120 binds to the CD4 receptor conformation change, bring the virus closer to the cell surface membrane, in which it binds to the CCR5 co-receptor as well, and that would also trigger a conformation change to the CCR5 co-receptor, and that would bring the viral envelope even closer, right, to the cell surface membrane. On top of that, there will be a conformation change to the GP41 as well, right, which will then facilitate the fusion of the viral envelope with the cell surface membrane. So as a result, the nucleocapsid that is within the viral envelope would be released into the cytoplasm. Okay, and obviously, as usual, right, the capsid coat will be broken down via uncoating, right, to release the content of the capsid coat, which would be the two copies of viral RNA, right, reverse transcriptase and integrase. Right, so within the cytoplasm, the reverse transcriptase would carry out the reverse transcription. It's a multi-step process. I'm going to show it to you here. Right, so this is the single-stranded, SS stands for single-stranded, right? Single-stranded viral RNA, which will be read by the reverse transcriptase, which will then copy a complementary single-stranded DNA, right? So now we have a hybrid, right, of RNA binding to DNA. After that, the reverse transcriptase would hydrolyze the viral RNA. So now we are left with a single-stranded DNA, which then becomes the template to synthesize the complementary strand, also by reverse transcriptase. All right, so multi-steps. The single-stranded viral RNA is now copied into a double-stranded viral DNA. Okay, so this viral DNA would then enter the nucleus and gets inserted into the DNA of the T helper cell, right? And this is catalyzed by integrase. So obviously the integrase need to get into the nucleus too to catalyze this reaction. Okay, the integrated viral DNA is now known as a provirus, not prophage, yeah, because this is not a phage, right? This is just a HIV virus, right? So provirus. It will remain silently in the T helper cell, but there will be no replication of or rather there will be no synthesis of any viral proteins, like no gene expression, right, of this pro-virus. Okay, so when the T helper cell that is infected and carrying this pro-virus divides, then as the T helper cell copies its DNA, the pro-virus is copied together because it's now part of the DNA, right, of the host cell. Okay, so from one infected cell, it will divide into 2 infected cells and so on. Okay, but when the infected T helper cell is activated, right, when there's an immune response, the T helper cell will start to transcribe its genes, okay, and part of it, right, or during this transcription of genes, the pro-virus will get transcribed as well. Okay, so when the T helper cell is activated, it needs to make proteins to do work, right, to trigger the immune system. Okay, so it will make protein, it will transcribe its own genes, but at the same time, the pro-virus is transcribed as well. Okay, so when the viral DNA, that's the pro-virus, is transcribed, it forms a viral mRNA, right? So this viral mRNA serves 2 functions, right? It obviously serves as the template for translation of viral proteins, since it's a viral mRNA, but it is also going to be the viral genome for the new viruses that are going to be assembled, because it's an RNA, it's a single-stranded viral RNA, right? So it will become the genome for the new viruses. Okay, so the viral mRNA will move into the cytoplasm just like normal mRNA, right? It will be read by the free ribosomes to produce the capsomeres and the enzymes, and it will be also read by the rough ER, right, to make the glycoproteins, right, because they are glycoproteins, right, so they must be made by the rough ER, get transported to the Golgi apparatus to be added with carbohydrates, and after that, right, the glycoproteins will leave the Golgi apparatus and get inserted into the cell surface membrane at exit point. Okay. As for the capsomeres and the enzymes, right, they remain in the cytoplasm, and in the cytoplasm we have the viral mRNA, right, which will become the viral genome, right? So the capsomeres and the enzymes, together with the viral RNA, right, will then form new nucleocapsids in the cytoplasm, right? So once formed, the nucleocapsids will move towards the exit point, where the glycoproteins are inserted. Right after that, then budding will occur. This is quite similar to influenza. The difference is there's no neuraminidase to facilitate the exit process. Right? So it's just normal budding, and the new viruses will be formed, and the viral envelope will be obtained, right, by the folding of the cell surface membrane of the T helper cells. Right, so that would be influenza, HIV.